So this is chapter 12, the eukaryotes, and this particular section is going to focus on both the protozoa and the helminths. So here are learning objectives for the protozoa. We're going to list the defining characteristics of them. We're going to describe the outstanding characteristics of the 7 phyla of protozoa discussed in this chapter and give an example of each. And we're going to differentiate an intermediate host from a definitive host. So protozoa are unicellular eukaryotes, which means they are one-celled organisms and they have a nucleus. They inhabit water and soil, and they have animal-like nutrition. They have very complex life cycles as well. And so feeding and growing, the feeding and growing form is what we call a trophozoite. An asexual reproduction is by fission, budding, or schizogeny, which is multiple fissions. And sexual reproduction is by conjugation. Some of them will produce a cyst to survive adverse conditions. So the paramecium you see here is undergoing what we call conjugation. And so conjugation is the sexual reproduction in ciliates, and each cell has two nuclei, a micronucleus and a macronucleus. The micronucleus is haploid and is specialized for conjugation. The micronucleus from each cell will migrate into the other cell during conjugation, and both cells will go on to produce 2 daughter cells. The condensed chromosomes are visible in the micronuclei. And so what they're going to do is this is how they get that genetic diversity that they're looking for, because they're both haploid instead of diploid. So the protozoa require a large supply of water. Many of them have an outer protective pellicle, requiring specialized structures to take in food. So ciliates will wave the cilia towards the mouth-like cytosome, and amoebae are going to phagocytize food. The food is digested in vacuoles, and wastes are eliminated through what we call an anal pore. So medicinally or medically important protozoa, so the feeding groups, the excavata ones. So if you think about excavating something, you might think of them excavating like a trench or a canyon or something like that. So we have the diplomads, the parabasalids, and the euglenozoa, the amoebae, and then the apicomplexa and the ciliates. So we'll talk about all of these. So the diplomads have no mitochondrion. They also have multiple flagella. The parabasalids, they have an undulating membrane. They have no cyst stage. The euglenozoa, these are the photoautotrophs that are facultative chemotrophs, and the hemoflagellates are transmitted by bites of blood-feeding insects. And so here we can take a look at a couple of these. So these are members of the superkingdom Excavata. And so the Clistomatics there in the top left, this one's found in the human intestine and can be mildly pathogenic. The cysts can survive for months outside the human host, and the fourth flagellum, the fourth flagellum is used to move food into the oral groove where food vacuoles are formed. Giardia, this is the trophozoite, and this one is an intestinal parasite and make you very sick. Giardia cysts, so this is the cyst form of that. The Trichomonas vaginalis, this is the flagellate that causes urinary and genital tract infections. The small undulating membrane here can be noted. And this flagellate does not have a cyst stage as well. The euglena, the euglenoids are autotrophs, which means they can make their own food, and the rings supporting the pellicle allow the euglena to change shape. The amoebae, they are going to move by extending their pseudopods. The entamoeba histolytica causes amoebic dysentery, and the acanthamoeba infects the corneas and causes blindness. Balamuthia is a granulomatous amoebic encephalitis, and so this one will kill you. So here we can see this, you can see the pseudopods up here at the top, and what they do is they stretch them out and then they kind of move towards it. So they have this like amoeboid motion is how they describe it. So they kind of reach out and then they bring the whole cell towards that pseudopod. Apicomplexa, these are non-motile, they're obligate intracellular parasites. They have very complex life cycles like Toxoplasma gondii and Cryptosporidium. The Toxoplasma gondii are transmitted by cats, and these are going to cause fetal infections, and so this is why they tell women not to handle cat litter when they are pregnant. The Cryptosporidium, this is transmitted via feces and causes waterborne illnesses. So this particular species here is some type of Cryptosporidium species, and this is the sporozoite in a fecal smear. So this can cause diarrhea, and it's not fun for anybody, but this is one of the most common causes of foodborne illnesses. So Apicomplexum, the Plasmodium, this is going to cause malaria, and they sexually reproduce in the Anopheles mosquito. And the mosquito injects the sporozoite into its bite, and the sporozoite undergoes schizogeny in the liver, where the merozytes are produced. Merozytes infect the red blood cells, forming a ring stage inside the cell, and so the red blood cells rupture, and the merozoites infect new red blood cells. So this is the life cycle of the Plasmodium vivax, the apicomplexum that causes malaria. And so the infected mosquito, we're going to start here at the top right here. And so the infected mosquito bites the human, and the sporozoites migrate through the bloodstream to the liver of the human. And they're going to undergo their schizogeny in that liver cell. Then they're going to be released into the bloodstream, and they can infect new red blood cells. The merozyte develops into the ring stage in the red blood cell, and the ring stage grows and divides, producing the merozytes. The merozytes are then released from the red blood cells when they rupture, and some merozytes infect new red blood cells, and some develop into male and female gametocytes. Another mosquito bite infects the human and ingests those gametocytes, and then in a mosquito's digestive tracts, the gametocytes unite to form a zygote, and this results in the sporozoites migrating to the salivary glands of the mosquito, which starts the whole process over again. Now, malaria has a very interesting kind of life cycle within the body. You'll go from kind of having a cyclic fever, and so when you do get infected with malaria, this cyclic fever, you'll feel like terrible. You'll feel really terrible for about 3 days, then it'll go away for a few days, and then it'll come back, and then it'll go away. So it's a very interesting kind of infection to treat, and it is treatable, but prevention is a very, very important part of the treatment of malaria. So ciliates, these are going to move by cilia arranged in precise rows, like the Balantidium coli is the only human parasite that causes, and so this causes dysentery. And so here you can see this, this particular one, this is Vorticella, and then the paramecium is there at the top. So it's covered with these rows of cilia, and it has specialized structures for ingestion called the cytosome. The one at the bottom here, this Vorticella attaches to objects in water at the base of its stalk, and so it's got that long stalk coming out of there. And the spring-like stalk can expand, allowing the Vorticella to feed in different areas, and the cilia surround the cytosome there. And so the cytosome is where the thing is going to eat. So this little ciliate is going to eat here. So either the paramecium or the Vorticella is going to use these kind of structures to ingest food. So here's your check your understanding questions. Please let me know if you have any questions. So next we're going to take a look at slime molds, and we're going to compare and contrast the cellular slime molds and the plasmodial slime molds. So cellular slime molds resemble an amoeba, and they ingest fungi and bacteria by phagocytosis, and the cells aggregate to form stalks and spore caps that differentiate into spores. And so here we can take a look at the life cycle of a slime mold. And so we start up here at the top here, so we're going to start right there, and the amoeba reproduces. Okay, the amoebae, they move towards the cyclic AMP signal given off by one amoeba, and they start to aggregate. And so then a sheath forms to create the migration stage, which is what we call a slug. And the slug stops the migrating and begins to form in a stalk differentiation stage. The fruiting body with spore cap forms, and the cells in the spore cap comes and they start forming, so that's this very large thing. And then the spore is released, and in that spore, the amoeba then germinates from the spore. And so this is the life cycle of a slime mold. Plasmodial slime molds have a mass of protoplasm with multiple nuclei. It moves as a giant amoeba. And so the cytoplasmic streaming, this is the kind of the circulation of things inside the cell. And so you might see a cell on a slide or something, and you're like, oh, there's something right here. Well, actually, when that cell was alive, it was moving around the entire cell. It might circulate around the outside if it was a chloroplast, trying to catch the best light. If it was something, that was going towards the nucleus, it'd go towards the inside. So things move around in cells. This is what they call cytoplasmic streaming. So here, this is the life cycle of plasmodial slime mold. And so the multinucleate plasmodium forms, and so this multinuclei can produce a lot of different proteins at the same time. And the plasmodium grows, distributing the nutrients by cytoplasmic streaming. And so you have these channels in here that are going to distribute these nutrients. And so the plasmodium separates into groups of protoplasm, and each group forms sporangia on stalks. And so then spores begin to develop in the sporangia, and the nuclear spores go through meiosis, forming gametes. When these spores get released, the gamete then germinates from that spore, and haploid gametes fuse, producing the zygote, and that zygote develops a binuclear division and cell growth, and then turns into a multinucleate plasmodium again. Now, one really interesting thing, and one thing you want to pay attention to, is the differences here between the sexual reproduction side and the side that's not sexual reproduction, the asexual reproduction. So it can continue to grow and essentially, continue to live by asexual reproduction, but it needs this sexual reproduction to get that genetic diversity. So here's your check your understanding question for the slime molds, and you need to be able to describe this to me for the exam. The helminths, these are worms. Helminths is a kind of an older name for worms because they have reclassified them, but this is a kind of a good way to kind of classify all of them together. So we're going to list the distinguishing characteristics of parasitic helminths. We're going to provide a rationale for the elaborate life cycle of parasitic worms, and we're going to list the characteristics of two classes of platyhelminthes and give an example of each. We're also going to describe the parasitic infection in which humans serve as a definitive host, an intermediate host, and as both, and list the characteristics of parasitic nematodes and give an example of the infective eggs and infective larvae. We're going to compare and contrast platyhelminthes and nematodes as well. So we've got two big phyla here. We've got the platyhelminthes, which are the flatworms, and we've got the nematodes, or nematoda, which are the roundworms. And so these are multicellular eukaryotic animals, and they're specialized to live in live hosts. They can lack a digestive system, they have a very reduced nervous system, they have reduced or lacking locomotion, and they have a very complex reproductive system. So dioecious, this is the separate male and female. Monoecious, this is where the male and female reproductive systems in one animal. And so generally, the life cycle goes from egg to larvae to adult. So here are your check your understanding questions. So make sure you guys take a moment to answer these. So let's take a look at some more of these nematodes, or not nematodes, but the helminths. So the platyhelminthes, these are the flatworms. The trematodes, or flukes, these are flat, leaf-shaped. They have a ventral and oral sucker, and they absorb food through their cuticle, covering them. The paragonimus is the long fluke, and the schistoma is the blood fluke. So here you can take a look at the anatomy. You've got the oral sucker and the ventral sucker, and you can see the intestine and the testes and the ovaries, so they are going to basically have both of them in there at the same time. The Clonorchus sinensis, so this is another example, so this is one of the ones that you might see. And so this is the Asian liver fluke right here. And so this is the has an incomplete digestive system, and heavy infestations may block bile ducts from the liver because it's trying to get that food. So here, this is the life cycle of the lung fluke, the paragonimus, any type of species of that. And so the hermaphroditic eggs, adult fluke, releases the eggs into the human lung. Okay, and so the eggs go through there, and the eggs reach the water after being excreted in the feces, and the miracidium develops in the egg and hatches from the egg. Then the free-swimming miracidium enters a snail, which is the intermediate host, and inside the snail, the miracidium develops into iridia, which is going to reproduce asexually to produce new ready egg. And so several Cercariae develop within that rhydia, and the Cercaria leaves the snail and enters the crayfish, and then the humans go and start to eat the crayfish after those cysts have formed to produce the metacarcaria. And so the infected crayfish is then eaten by the human, and it develops into the adult fluke. The cestodes are the tapeworms. The scolex is the head that has the suckers for attachment, and they absorb food through their cuticle. The proglottids, these are body segments. They're going to contain male and female reproductive organs. And so you can see the differences here. You can see that they're very similar in kind of design. At least their reproductive sections are very similar to the flatworms we just looked at. And so you can see the hooks and the suckers on the scolex, and then the proglottids. You can see how developed they are. The cestodes, they use humans as definitive hosts, so the eggs from the proglottids are ingested. And then they hatch into larvae and bore into the intestinal wall. They produce what they call cysticerci, and the Tinea solium is the pork tapeworm, which is the one that probably most commonly infects humans. So eggs are ingested and hatch in the intestine, and the larvae can also migrate into the liver or lungs and develop a hydatid cyst, and Echinococcus granulosus is an example here of how this happens. So here's a diagram of the life cycle of the tapeworm. So the adult tapeworm releases the eggs, and the human ingests the immediate. If it ingests the eggs, it doesn't go through, so it stops here. Okay, but the intermediate host is where it is going to go through, and it's going to develop into that tapeworm that can actually infect the definitive host. And so the intermediate host is where some type of reproduction takes. So the eggs hatch and they migrate to the liver or the lungs, and the larvae develop into hydatid cysts. And so the definitive host then eats the intermediate host. This is the wolf here, and so ingests the cysts, and it gets the tapeworms, so the scolices from the cyst attached to the intestine, and they grow into adults to create more eggs. So here is your check your understanding question. Please make sure you take a moment to take a look at this. Roundworms, these are generally cylindrical, hence why they call them round, and they have a very complete digestive system. Dioecious, the males contain spicules, and they're free-living and parasitic. Eggs are ineffective for humans, so the Ascaris lumbricoides infects the human intestines. The Balascaris procinus is the raccoon roundworm. Trichuria is the whipworm, and the Enterobius vermicularis is the pinworm. And so here we can see a little bit of the anatomy of these things. The males are generally a little bit shorter than the females, and so they do reproduce asexually, or they do sexually reproduce, excuse me. So the larvae are infected for humans, so Strongyloides, these are a re-emerging infection. The Necator americanus and the Aclystoma duodenale. These are hookworms. They enter the skin and are carried to the intestines. The Diphtheria immitis is spread by mosquitoes, and it causes heartworm. So you might give your dog, or you should give your dog, heartworm prevention every month, and this helps to limit the spread of that disease. And so here is a heart that has been infected by the heartworm, and you can see how terrible this could be for the dog. So a lot of times when the dog does have heartworm, there's nothing they can do about it, and it does severely limit their cardiovascular capacity. So here's your check your understanding questions. Make sure you guys take a moment to take a look at these. So arthropods as vectors. So we'll talk about this. Arthropods are animals with segmented bodies, hard external skeletons, and jointed legs. Vectors are arthropods that carry pathogenic microorganisms. And we have some representative classes. We have arachnids, which have 8 legs. We have crustaceans that have 4 antennae. And we have insects that have 6 legs. So a mosquito has 6 legs, but this tick has 8. So 6 and 8. And so this is one way that you tell them apart. And so here's some other ones. So the human louse, so the head lice is caused by this. This is the same louse that you find in what we would commonly call crabs, but pubic lice. And so this has 6 legs. The rat flea also has 6 legs. The deer fly is going to have 6 legs, and the kissing bug is also going to have 6 legs, because all of these are insects. And so what they generally do is they have some type of transmission. So they have mechanical transmission or biological transmission, where the pathogen can multiply in the vector. And then the definitive host is the microbe's sexual reproduction takes place in the vector. And so we've already covered that when we talked about the tapeworm, how it had a definitive host versus a intermediate host. And so here's your check your understanding questions. If you do have any questions, please feel free to ask me and let me know.